Hi everyone. Today with this video, I'm going to let you know about how PCR will help to detect uh, disease-causing microorganism. Today, I'm taking one example of Escherichia coli that is normally found in water, drinking water. So, for the detection of E. coli, normally we will take a sample from contaminated water like rivers or from other sources. Afterwards, we will perform a DNA extraction using CTAP method or even the physical method. After extraction of DNA, we need to have like a primer of E. coli. Uh, so we will design primer and obtain from the relevant sources. Then after preparation of a master mix, uh, and primers along with the sample of E. coli, we will amplify that gene in PCR. So after amplifications, we will have the sequence. Uh, if we have like a sequence multiplied or quantified more than the initial concentration, after a 30 to 40 cycle, we can easily understand that the gene of E. coli is present there. So based on if there is for the detection of whether the E. coli is present or not, after PCR, we will do gel electrophoresis with 1% of agarose, and if there is presence of a band, then we can understand that DNA band is absent. It means E. coli is absent, and if there is presence of a band in the uh, or prepared PCR result, we can understand that E. coli is present. It means PCR method is faster method than culturing bacteria, which can take like 24 to 48 hours, and further biochemical experimentation for detection. And PCR is a reliable method because we can identify the organism based on their genes. Likewise, for genetic mutation, um, we can use PCR. PCR help to detect a mutated gene either in human or in other kinds of diseases. For that, normally what we'll do, we will have like a DNA sample uh, from the patient blood or from any other sources. So then we will amplify the region of DNA like CFTR gene that causes due to mutation CFTR gene. Uh, this is the reason where this normally mutation occurs. So mutation analysis shows that DNA amplified DNA sequencing through bioinformatic analysis, allele specific PCR restriction enzymes. So in interpretation, normal gene has no mutation, but altered gene sequence presents mutation. So doctor use the information to diagnose genetic disorder or carrier. And for forensic identification as well, PCR is extremely helpful and useful because it can amplify very small DNA sample, uh, normally possible from what DNA sources like hair, blood, saliva, even skin, scalps. So, uh, crime scene investigation, what they will do, like investigate, try to find a tiny bloody stain at a crime scene, or through the DNA extraction, or what will happen? DNA is extracted from blood initially, then there will be PCR amplification. So through the specific DNA marker like short tandem repeats, these markers vary between the individuals and also represent a unique identity of individual organism. So DNA profile creation will normally helps to form a DNA fingerprint. So based on the DNA profile, it is compared with the suspect DNA. If the STR pattern matches with any suspect may have been present, we can diagnose on such a way. So PCR made a modern forensic DNA analysis possible. Likewise, for gene cloning in biotechnology, it is also useful. Normally, we will produce this useful protein. So for that, initially, what we will do, extract the DNA from either from microalgae or other cyanobacteria and amplify a gene with the PCR techniques to produce like a desired protein uh, and carbon capturing gene. So we insert that gene into the plasmid and we create a plasmid vector. Afterward, afterwards, we transmit that plasmid vector into the host organism like a bacteria, like E. coli, which multiply very fast. So after that, those organisms are responsible for the production of a protein in large amount like insulin, enzymes, vaccine, industrial protein, and we recovered that from that organism. Thank you so much for this video. I will hope that this video will make you clear about the PCR uses. We will meet in next video. Stay tuned.